Science is a noble pursuit. Experiments in the lab are adventures, but they can get tedious as you experiment and tinker, then tinker some more. But when something works and it's quite a big deal, that's cause for celebration. This year, it's 2026. It's been 20 years since research was published on how to generate induced pluripotent stem cells. These cells are special. All cells in our body have special duties in the brain, muscles, the gut, eye, everywhere. They aren't born specialized, they become specialized. What if, scientists have long wondered, you could turn the clock of development back to the time when the cells weren't specialized yet, and they could become any type of cell. That's when cells are pluripotent. This achievement happened 20 years ago, and it happened in Kyoto, Japan, at Kyoto University's Center for Induced Pluripotent Stem Cell Research and Application. In the summer of 2005, Dr. Kazutoshi Takahashi ran to his mentor's office, Dr. Shinya Yamanaka. He had to urgently show him something. That something turned out to be cells in which the clock had been turned back by treating them with only 4 specific proteins. The scientists published this work in a paper that was in 2006. Many labs had been working on this, trying to find ways to turn back the clock in cells. 1962 was an important study by Sir John Gurdon, and there have been many, many more. And this is only an incomplete timeline. Dr. Gurdon and Dr. Yamanaka received the Nobel Prize in 2012 for their achievements. Dr. Gurdon passed away in 2025. Time to ask Dr. Yamanaka how he is celebrating this science birthday.
Usually we have a cake, like strawberry or chocolate cake, and we put candles. For example, this is a 20-year anniversary, so if possible, we put 20 candles and light them all on, and then we blow it off.
So I just wanted to also make sure I understand how many labs you run. So you were director emeritus of CIRA, I think. You have a lab there, though, at CIRA, and you have a lab at Gladstone. And I think you are director of the CIRA Foundation. Did I understand that right?
You are right, but my main lab is now in San Francisco. I have like 10 people there. I only have one research associate here in Kyoto at CIRA. So my main activity is in San Francisco. As for CIRA Foundation, I am the president. But we have, my role is only part-time at CIRA Foundation.
This year, the very first 2 therapies that use induced pluripotent stem cells were approved by Japanese authorities, one for Parkinson's and another for heart disease. I should say conditionally approved, since there will be monitoring for 7 years.
May I ask, now talking about stem cell therapy, which is obviously one of the greatest applications of this big discovery, right? When you started working on the iPSL project, was your vision therapeutic to begin with?
Yes, exactly. So when we were working on the generation of iPS cells, I had stem cell therapy in mind.
Wow. And how come you did not, I mean, I'm very glad that you did that, but how come you did not turn to something that was already out there? I mean, I don't know, something drug-based, for example. What did you see more in the field of reprogramming in terms of how it can be therapeutically translated back then?
Well, many diseases are caused by a loss of just one single cell type, like Parkinson's disease, liver failure, heart failure. So if we can generate a huge amount of that one kind of cells, we should be able to help those patients. And ES cells, embryonic cells, were the answer for that. However, ES cells have a huge hurdle, ethical hurdle. So my motivation to generate iPS cells was to overcome that hurdle of ES cells.
The group that has managed to make clinical grade iPS cells, I mean, the effort is so tremendous. And I know that it's possible for you to give advice to many, but what do you think is the biggest thing that people underestimate when they do that? Is it that some cells that you have aren't in the cycle that they need to be? Differentiation hasn't worked right. What are some of the things that people underestimate in this task of you can't just passage the cells and go, okay, you're ready for the clinic, right?
Well, moving from bench to clinic was much more difficult than I thought. I thought it would be 10 times more difficult to generate clinical grade iPS cells than making research grade iPS cells. 10 times more difficult. I was wrong. We learned it is 100 times more difficult. So many reasons. It's a combination of many factors. But moving from basic science to clinics is really difficult. Many people do underestimate that difficulties between bench and bedside.
And do you think this is where the major difficulties in the field will be for the years to come? I mean, as an editor, I still continue to see a lot of manuscripts on reprogramming. Many of them great, I must add, but mostly focusing on refining the protocols or providing adaptations, not necessarily though changing our view of things in the field of reprogramming. So I guess my question is, do you think the major breakthroughs to come now are going to be on the application side in the clinic, for example, or do you have burning questions in mind in terms of the reprogramming field, like as a process itself, for the years to come?
Both are very important. Reprogramming is still, I have to say, it's still like a black box. Especially, we still see a huge degree of variations among iPS cell lines, even from the same donors. So we really want to have a better way to generate iPS cells with uniform and a very high quality. That's what people at CEDAR and at many other places are still trying to achieve.
This heterogeneity, is this a strength of these cells maybe, and not just sort of a problem, or maybe it's actually something that has to be solved because they're not identical, obviously?
Yes. So right now, many applications, many companies are trying to focus on just one line, allografts, not from patients' own cells, but from healthy donors. So in that case, we have enough time to select the best line. And in that sense, heterogeneity may be good because we can select the best line from many different iPS cell lines. But I believe the ideal usage of iPS cells is to make autologous iPS cells from each individual patient so that we can avoid the usage of immunosuppressants. For that purpose, we really want to avoid heterogeneity. because in order to achieve autologous iPSL therapy, the best scenario is we just make one iPSL line from each donor, and that line works well. That's the scenario we want to see, and for that purpose, we want to avoid any heterogeneity.
I was looking at the paper also about the fact that you were surprised that a nanog wasn't needed. And I thought that nanog is kind of like a pluripotency, powerhouse, but I guess it isn't. Or is basically the knowledge about all of this settled? Or are there things for PhD students and postdocs to still work on in terms of maintaining pluripotency?
Yeah, it was very surprising for us to know that nanog is not among those 4 factors. We are one of the 2 groups who reported nanog for the first time in 2003. So we are hoping that nanog is absolutely required. So now I believe that nanog is more like a safeguard for pluripotency. It's not the determinant of pluripotency. So nanog is still important, but in order to induce pluripotency, we do not need nanog. So science is interesting.
I just wanted to ask this other thing about the name, if I may. The embryonic stem cell-like. I know that you're very careful with language. So it's almost indistinguishable, but yet you say it's like. Because of the provenance only, or are there other subtle, very differences between embryo?
It's a good question. I believe there was a paper from Konrad Heckendringer lab showing that if you generate ES cells and iPS cells, genetically identical ES cells and iPS cells, they are actually indistinguishable. So I think we can say ES cells and iPS cells are at least nearly indistinguishable, but at the same time, we do see more heterogeneity or variations among iPS cell lines. So if you pick up the best iPS cell line from each donor, I think they're very good. But at the same time, we could have not so good iPS cells from the same donor. So we still have to select a good one for your own for your own purpose. I believe that's where we are.
And I wanted to sneak in one mentoring question, and then Stelios, you should go ahead. So I watched some of your talks where you talked about vision and hard work. You also mentioned what it was like to come back to Japan, and that people were critical of your articles. When people like a postdoc are in that phase, how do you motivate them these days? Whether they went to the US or not, stayed in Japan, or maybe they always stayed in England or wherever, how do you motivate them to keep going on this? Because it is very hard.
Exactly. But you should enjoy science. I started my career as a surgeon, as a clinical doctor. Then I moved to science. There is a huge difference between clinical work and basic science. In clinical work, you cannot enjoy failure, unexpected result. But in basic science, at least I enjoyed a lot about my unexpected result. That's the belief of being scientist, especially in basic science. You can enjoy unexpected result. You can enjoy, in a sense, you can enjoy failures, and you could learn a lot from unexpected result, and you could learn a lot from failure. and enjoy, be en- you have to enjoy.
I was a researcher myself, so I know all about the enjoyment. I also know a big chapter in research is doubting, right? So let me take you back to 20 years ago. The moment, I mean, obviously we knew already that you can take a somatic cell and reprogram it, right? But with four factors, that was the big thing in it, right? So the moment, the first time that you saw the results, that it was possible to use those factors to reprogram a somatic cell, did you believe it immediately? How did you react to it?
So it was summer in 2005 when Kazu Takahashi ran into my office and said to me, Dr. Yamanaka, I can see a colony, ESL-like colony in microscope. You have to come with me. And I looked at the microscope and I saw multiple colonies that just looked like ES cells. But I told Kazu, don't get excited. I'm sure this is a result of contamination. So I'm 99.9% confident that this is a contamination. So you should just repeat the same experiment again and again, and he did. And every time he tried, it worked. So that was how it happened. So there was no moment that we got super excited. At least I was very, very careful, and I was very nervous.
And how about others? Like, did you also? I'm sure you got a lot of people excited, but after the paper came out, until there were the first papers that really recapitulated what you found, did you also meet people doubting a little bit the fact that four factors and that's it, you can program the cell?
Yeah, after actually when we tried to publish that first work, we were very nervous. We knew we were going to get huge criticism. So we were very afraid of it, and that was one reason why I made only two authors on that paper, because we knew we will be on a huge criticism. We had to go through all of those criticisms. So I think only Kazu and I should receive those criticisms. And it did happen. We got many skepticisms and criticisms. So when we saw a paper from Dr. Yanishi's lab within a year, reporting that they were also able to recapitulate our work. And when I learned they use the same name, iPS cells, we were so happy. Well, it was a start of fierce competition, but nevertheless, we're very happy to see their paper in Nature, actually.
In 2046, first question is, what will you be running in marathon-wise? What will your time be? What do you think in 2046? And the second question is, what do you think stem cell biology is going to be, Sierra or elsewhere? Where is it going to be, particularly in terms of the clinical translation? So marathon and 2046, what is the world going to be like?
So 20 years from now, I will be 83. So I hope I will be still running marathon. As for the time, in 20 years, Uh, so my time is still getting better and better. So according to this trend, I should be able to break 2 hours in 20 years.
Wow. Oh, cool. That would be great.
But I need to rejuvenate myself. But I hope I can still run. Yeah, but I just hope I can still run marathon.
Yes,
20
we'll
years from
see.
now.
We'll see you then.
We'll see.
And
We'll
so
see.
in
iPS cells may help.
Yes, I was wondering about that. So do you think that stem cell biology, particularly iPS cells, will have made their way into the clinic in many ways in 20 years? And I know this isn't fair as a question, but I'm just curious what you think 20 years will lead, because 20 years have really done a lot. My goodness.
So many clinical trials are ongoing in Japan, in US, in China, and in many other countries. So in 2046, I hope many of them will be approved by then, so that many patients can receive those therapies using iPS cells.
Thank you so much, Dr. Yamanaka.
Thank you.
It was a real pleasure.
My pleasure. It was fun. Thank you very much. Thank you.